Hello, everyone. This is SBI, and today we're interviewing Dr. Califano of the Systems Biology Department, who has done extensive research in transcriptional regulatory networks and has applied them to a variety of fields, including tumorigenesis and cancer diagnostics. Really amazing research. And here we're going to interview him and ask him a few questions about his research and his perspective on systems biology and even how that can be expanded to synthetic biology as well. So our first question today for you, Dr. Califano, is tell us a little bit about yourself. Like, who are you, how you became involved in systems biology, and how you ended up here at Columbia. From your website, it says that you were one of the founders of the systems biology department here. So can you also describe how that came about as well?
Sure. So first of all, great to be on your podcast. I am a physicist by training and also have profound attention deficit disorder, so I can't do the same thing for more than 3 years, and that's how I got from physics initially to computer science when I was working the first few years at IBM Research about 30 years ago. And then at one point we discovered that many of the things that we were doing in computer science were actually extremely relevant in the context of the nascent field of computational biology. And that led me to sort of create a first group at IBM that then became a large-scale centers across 4 continents for computational biology. And one of the reasons that I then decided to move to academia is that within that context we could do a lot of computational work, but we couldn't do the experimental work that was necessary to actually inform the analysis that we were performing. And so the great thing about being at Columbia is that I was able to then start my experimental lab. And as a physicist, I essentially really think about biology in a way that is a little bit orthogonal to the way that is generally used by regular biologists. That is, we look at it instead of as a black box, we want to open the box and really understand very precisely how it works inside. And so this has been done usually for individual genes and or individual gene products, trying to understand how a particular protein interacts with another one, but has never been done, at least at the time, had never been done on the scale of what we had in mind, which is to understand the full set of regulatory interactions, signal transduction interaction, post-transcriptional interactions that actually determine the cell behavior. And so this is building that map of molecular interaction that determine behavior was our first goal. And then basically once we achieved that, we decided to now how can we effectively use that information to ask relevant questions about the cell. So you asked me about the Department of System Biology. My goal since coming to Columbia has always been that of creating an environment where the experimental throughput biology and high throughput biology down on the experimental sides and high performance computing down on the sort of the modeling and computational side could be merged into sort of a seamless and framework. And this was done initially to the creation of what we call the Joint Centers for System Biology, which was the Columbia Genome Center plus the Center for Computational Bioinformatics, and Computational Biology and Bioinformatics that I started with Barry Honig. And so that worked out very well for a while. And the reason we then decided to move it into a department is that we started realizing that a number of critical people we wanted to recruit to Columbia were kind of falling through the cracks in the sense that there was no department that really represented a home for them. And unfortunately, appointments have to be made in departments. And so we lost a couple of really great opportunities of people that actually, researchers that ended up really building major labs and becoming some of the most well published in the field. And so that sort of drove us to then deciding that this was the appropriate time to actually create a department that would cater directly to the interest of people like that, and that has been very successful. So I'm the chairman of the department, and so I founded it in 2010 with Barry Honig, and sort of Barry and I have been sort of running this operation and trying to extend it as much as possible as an environment where you can do collaborative research.
Inspiring. But one follow-up question to that is, like, in your perspective, how would you, like, for our viewers, how would you define systems biology? And especially maybe it would be like just in contrast to synthetic biology. Like, how would you define? What's your
Right.
definition of systems
So
biology?
if you ask 10 different people, you get 10 different definitions of systems biology. And the reality is that this is not completely unwarranted because it really depends on the focus and on the slants that you put on the discipline. The way we interpret system biology here at Columbia is essentially as model-based biology. We think of system biology as the natural transition of an empirical science into becoming more and more model-based and more analytical-based. So, for instance, there are many places that do what we call integrative biology. They combine together data from many different data sets. For instance, you could take metabolomic data and genomic data and gene expression data, and then they integrate it, but in doing so, they still integrate it kind of one gene at a time, right? And so they can do it very fast. They can do a massive environment. They can do it, for instance, by using now CRISPR-Cas9 libraries, pool libraries to perturb every single gene in the genome. And many people think of that as potentially being systems biology. We don't think of that as system biology. We think of that as integrative biology. What we think of system biology is when you actually first build a model of how a particular biological phenomenon should work, and then you use that model to make predictions that they can bring to the lab and validate mechanistically. So this is done not just in the area where I work on, for instance, which is regulatory and signaling networks, but it can be done, for instance, in labs like Barry Honig's lab, where they use very sophisticated sort of angstrom-level models of a protein structure to actually predict protein-protein interactions, and then be able to go to the lab and say, oh, look, we predicted 100,000 or 1 million protein interactions, and now we can validate them in the lab. To the lab of Rahul Rabadan, where he used a very complex algebraic topology model to start trying to understand how mutation essentially evolve within a particular patients as they undergo progression in cancer. So the models can be very different. In my lab, we use, as I said, regulatory models. So we say, okay, these are the interactions that determine cell behavior. So now if I see this behavior, what is the gene or genes or gene products that cause it? What are the drugs that we can use to counteract that effect? But as I said, this is not the only way to do it. There's almost every single lab here, and that's what we like about the team of faculty that we have assembled here is that almost every lab has a completely different and orthogonal approach, but they're all based on this idea of having an underlying model. So if you want to put this in context of other sciences, almost every science that is now considered to be a natural science, with the possible exception of biology, which is undergoing a transition like now. But if you think about chemistry, you think about astronomy, meteorology, everything. The science has undergone a fundamental transition and essentially has experienced what its golden age, if you want, when it moved from being based on a fully empirical base to becoming driven by analytics, and then using the analytics to make prediction and going to the model. For instance, in physics, if you were in the 1200s and you consider yourself a natural scientist, you would potentially observe and time a ball rolling down an incline and try to measure, take all the measurements. After Newtonian mechanics came into play, all of a sudden you don't have to do those observations anymore. You can actually predict it very efficiently just by knowing the angle of the incline, and that's pretty much about it, and the time you're observing the ball rolling down. So there are certain aspects, for instance, virtually all particles that have been experimentally validated in particle theory have actually been first predicted computationally. I was very intrigued by all these reports about the discovery of gravitational waves. They were not discovered, they were just validated. I mean, Einstein postulated them and essentially discovered them from a purely theoretical approach, and then finally it took a while for the experimentation to catch up, because the devices that are needed to detect those type of things are extremely sophisticated and complex, and it's a major achievement, well deserving the Nobel Prize. But it was not the discovery, it was actually the validation. So in biology, I think we're undergoing that transition right now, and what system biology is trying to do is to create this framework that will allow a more and more analytical approach to being able to predict the behavior of the cell.
That's something that's aspiring for researchers today, given this day and age, and the technology that we have. Modeling does play a very essential, and it's paramount to the research we're doing now, especially for empirically verifying our data. So it's just really notable of how systems biology is applying modeling to biology, and I think it's going through an evolutionary stage right now.
So I know that your lab has recently published a number of papers on transcriptional regulation in various types of cancers, and as well as in the past in metabolic networks. Um, but can you tell us about how this research ties into the larger work of cell regulatory networks, your current research?
Sure. So we start from taking a slightly different view. So instead of starting from the infinitely small and trying to understand how it works one little piece at a time, we try to start from the larger picture. And one of the things that, for instance, people have not considered is that if you were to imagine the cell as being simply a bank of switches, 20,000 genes, 20,000 on-off switches, okay? That cell, and this is the simplest possible model, obviously much more complicated than that, but this is the simplest model you could create. Now, that cell would have 2 to the 20,000 possible states, okay? I don't know if you know how many atoms are in the universe, but there are about 2 to the 400. So this is a number that is almost infinitely larger than the number of atoms in the universe, which is pretty large already. So one of the questions is we started asking is, okay, why do we need this complexity? There's only a few thousand states that the cell has to be in, okay? So why do we actually need 20,000 switches to make the cell work? And one of the reasons is that the cell has to essentially be extraordinarily redundant and resilient to any kind of external input that may go wrong. Your temperature, yesterday was 75 degrees, today is 40 degrees. A cell that wouldn't have method to compensate for that would die. And so about 90% of the cellular machinery is actually involved in performing a function that's called homeostatic regulation. It's maintaining the state of the cell independent of all the perturbation that the cell is being subjected to, both endogenous and exogenous. So we started from that perspective, and we asked the question, so how is it possible that cancer cells are incredibly tightly regulated? They're as tightly regulated as normal cells. Basically, if you take 100 cancer cells from 100 different patients with the same tumor subtype, or 100 normal cells from 100 individuals from the same tissue, they are as tightly regulated. Okay, they're not different. They just sit in a different space of those 20,000 switches. And so the question is, how is it possible that you have exactly the same transcriptional state for these cells and they do exactly the same thing, and yet the genetics that induces those states are completely different? Now, there's no two patients with the same set of genetics insults, and these cells, regardless of what genetics insult, end up being the same type of cancer. And so we thought that the only way to answer that was by getting an intimate understanding of what happens in terms of regulatory processes, because one way in which this could happen, and we had a hint of that in a paper that was published in Nature many years ago with collaboration with Laura Pasqualucci and Riccardo Della Favra, that in fact this mutation converge, canalize on activating or inactivating a very small number of proteins, and then it's the aberrant activity of those proteins that induces the state of the cell. And so we spent the last 14 years building the regulatory maps that were precisely mechanistically validated to a large degree. So about 70 to 80% of what is in our maps is actually experimentally validated in the lab. And then using those maps to try and figure out how, in fact, that canalization occurs. And that led to a very, I think, fundamental discovery, which is that cancer states are really regulated by only a handful of proteins that we call master regulators. And these master regulators work together in typically very small modules. We're talking about 10, 20 proteins. We just published, this is in press now, a 10 protein module that regulates, for instance, neuroblastoma. And what happens is that these modules really operate as macroscopic on-off switches. So they can't be in a state that is an intermediary state. They can be either on or off because they're very tightly regulated. They're like an air conditioner in a room. They have feedback mechanisms that tell them when to turn off and when to turn off. And in cancer, they are turned on, and what we need to do is actually turn them off using drugs or other methodology. And so this sort of explains, first of all, why these cancer states are so discrete and actually quite distinct one from the other. And yet many, many patients with completely different mutation end up having very similar cancer state. And number two, the fact that when we actually are drugging the oncogene, the oncoproteins that are sitting upstream of those modules, we're actually doing something that is counterproductive, because what you're really doing is, and we recognize that right now this is the best that we've been able to do, but what you're doing, you're simply selecting for subclones within the tumor that have mutations that are either bypass or alternative mutations to the one that you are targeting, and they still impinge and activate the same regulatory modules, and so the tumor will relapse and relapse in a form that can no longer respond to the original drug. And so that's why we see that all these targeted inhibitors, essentially don't cure cancer, with very, very few exceptions. They simply essentially slow it down, and then they produce response. What we suggest we should do is actually drug this much more central mechanism, if you want, the core engine room of the cancer cell. And by doing so, you're essentially, the only thing that the cell is able to do is to literally reprogram itself into another state. But there's only a very small number of states that a cancer cell can be in, typically 2 or 3. As opposed to, again, larger the number of atoms in the universe, possible mutational patterns that can induce resistance to a particular drug. So you're going from something that is an infinitely large set of potential solutions to something that is very discrete. You have 1, 2, 3 possible states you can have, and if you figure out how to fight all of them, then at some point we may be able to defeat this disease altogether.
You've been speaking a lot about how you build these transcriptional maps and look at all these interactions among different transcription factors. So how do you measure those things, and what tools do you use to analyze the data and figure out, say, what those master regulators are?
So that's a great question. Actually, this is to some extent where I reconnect with my physics background. We use a branch of physics and mathematics called information theory, which essentially postulates certain things that are especially relevant in the context of dissecting regulatory networks. For instance, one of our critical algorithms that we use to reconstruct regulatory networks from gene expression data is called Arachne. And what Arachne does, it plays the telephone game. So you remember when you were kids, somebody would speak in the ear of somebody else, and there was a long chain, and at the end of the chain, the message was all scrambled. Right? This is exactly what happens in biology. If the information is transferred indirectly through a series of events, then that information a part of that information is lost. But if you transfer it directly, you lose a little bit less of that information. So if you can measure information on every single path, both the potentially direct one and any one that goes to any other intermediary, and you find that the potentially direct path is the one that transfers the highest amount of information, then you know that that is potentially a real biochemically and physical interaction. And so what this has been really surprising, this is a paper we published in 2004 or 5, if I remember correctly, again in collaboration with Riccardo Dalla Fabra, where we were the first lab to generate a complete regulatory map for a human cell, in fact for any mammalian cell, and we will show that there was extremely high validation rate, and throughout more than 70 papers that we published on this particular subject, we've shown validation rates to be in the order of 70 to 80% if you go to the actual lab and you try to biochemically validate that these interactions are correct. So once you have this map, it's a little bit like shooting fish in a barrel, because the most important thing in disease, and it's not just cancer, we've applied this to neurodegenerative disease, so now we're doing work with Domenico Chili on diabetes. We've used it even for neurobehavioral disease like alcohol addiction, for instance. And where it works actually the best is in reprogramming, tissue reprogramming. So in, to some extent, what we call regenerative medicine or tissue engineering, where we recently published in collaboration with Michael Shen Lab and Koryabata Shen Lab the ability to reprogram cells from one tissue type to another tissue type, in that case fibroblasts to normal prostate epithelium, by just targeting a very small number of proteins that we call master regulators of that transition. So the master regulator are simply the proteins that regulate the genes that are differentially expressed between the two states. And so if you know the differential expression of the two states, and you know what are the targets of each protein in the cell, it is almost obvious how to find the master regulators. And we designed an algorithm called Viper that was published in Nature Genetics a couple of years ago, that allows you to do that using even single samples. And it's particularly useful, for instance, in single cell analysis, because in single cells you have an enormous number of dropouts, that is, genes for which you don't see even one read in a gene expression profile. And because Viper uses all the targets of a protein to predict its activity, now you're averaging over such a large number of genes, that some of them will have sufficient level of expression, so you can now measure the activity of proteins even from single cells. So this is kind of gives you an idea of how we use it. We then use something called the conditional mutual information, which is another information theoretic approach, to go upstream of the master regulators and find out all the proteins that can modulate their activity. And guess what? Those are the proteins that contain the mutations. And so it's a paper that we published in Cell showing that you can literally systematically go upstream of the master regulators and find all the mutations that had been missed. And you can find dozens of mutations per patient that are upstream of these master regulators and not upstream of other transcription factors. So that's kind of gives you an idea of the kind of approach that we use.
And in recent years, you've been taking the information you glean from these sorts of analyses and experimentation, and you've actually been applying it in clinical trials. So can you speak to how that effort has been going?
Right. So obviously, the proof of the pudding is in the eating, and the ability to reconstruct regulatory networks is only sort of exciting to the point that it allows you to make predictions that then can be used effectively. And when we're in a medical school, so one way to use them effectively would be in trying to figure out how to better achieve patient response, for instance, in cancer. So the idea is quite simple. The idea is that once you know what the master regulators of a particular tumor are, and we can do that literally within seconds of receiving the gene expression profile of a biopsy of a patient. Now the question is, how can we reverse the activity of those proteins? Right? How can we shut down the proteins that were aberrantly activated, and how can we rescue the activity of the ones that were shut down by the tumor mutations? And this can be done very efficiently because we developed, in collaboration with Peter Sims, a technology called Plate-seq that allows us to do RNA sequencing for about $20. And that means that we can now afford to do perturbational assays, and we've done about 20,000 of them so far, where you take a cell line or an organoid or even a mouse where you have the complete recapitulation or very high recapitulation of the master regulators of the patients. And now you can perturb that cell line with drugs and ask what happened to those master regulators after the drug is put in, and use exactly the same algorithm. Use the Viper algorithm to measure the activity of the proteins in the patient. So you can now use the Viper algorithm to measure what happened to the activity of those proteins after you put the drug in, right? And so this is essentially almost like literally a key and lock mechanism where we use the small master regulator module, about 25 and 25 positively and negatively regulated master regulators, as a gene reporter assay to actually understand whether the drug has an effect on them or not. Okay, and so what was really remarkable is that now we have about 7 clinical trials that have either already started or already funded and about to start, and some of them have already moved to phase 2. For instance, one for HER2 positive breast cancer that failed to respond to trastuzumab. And the idea is that we simply can either identify this mechanism a priori and find out what is the drug that gives you the best opportunity, and then use a traditional clinical trial, either with a drug or a drug combination, and we have a few of those, or even more sophisticated, actually take each individual patient from a variety of different tumors and just on the spot identify which drug would revert the master regulators, and then we use that drug in a transplant of that patient in a mouse. And just to give you a sense, right now we've tested 34 drugs that were predicted computationally using this approach in patients that have progressed, so in a way that was not responding to any drug, about 3 to 7 different lines of therapy, and 20 of the 34 drugs, so about 60% of them, have induced actual what we call objective response. That is either stable disease, so that instead of growing exponentially, the disease flattens out, or remission, so or partial response. So this is quite promising, and we had some anecdotic responses in patients. So now we are doing, for instance, in collaboration with Ken Olive and Gary Schwartz, we're doing a trial for pancreatic cancer that was funded by the Lustgarten Foundation, where we're applying this methodology to pancreatic patients that are predicted to respond to a small numbers of different drugs. So we have a repertoire about 8 to 10 drugs that will be put on the study, and each patient will be assigned to the drug that will be the best possible match for their specific type of pancreatic cancer.
So your work and research can be applied to precision medicine, um, in targeting these master regulatory nodes in specific diseases that specific people have. But precision medicine is generally very expensive, and not scalable to the general population. Are you taking any steps to make it more accessible for future therapies?
That's a great question. So first of all, let me just make a comment that we use the term precision medicine, but actually it is not very precise at all so far. In fact, we know that many of the patients that have so the precision medicine is now predicated on two type of approaches. One is targeting mutated oncogene. The other one is using immunotherapy approaches. In both cases, we really don't have a good grasp on either why patients respond and why patients do not respond. And so what we call precision medicine, the precision is simply in the identification of the potential target, but then there's a missing link between that target and what is the actual response or phenotype of the patient. What we're trying to do is to actually build the connective tissue between this observation and the actual result, so that instead of having just a statistical association, we can actually predict it in a much more deterministic way, right? And so really trying, if you want to put precision in precision medicine. So you're saying that this is expensive. I mean, one reason it's expensive is because the analysis of a genetic profile of a tumor is still quite expensive. You know, we're talking about maybe $1,000 now, but $1,000 for research purposes. So if you have to do it in a way that is completely reproducible, it's called CLIA, New York State CLIA certified, then that cost goes up quite significantly. And even panels of genes that are now adopted. So for instance, Columbia has a 500 gene panel, Foundation Medicine has a 400 gene panel, etc. Even those panels are quite expensive, several thousands of dollars. But that is compounded with the fact that only about 10% of the patients that are screened using those panels actually have an effective, what's called PFS, or progression-free survival improvement because of the treatment. Okay, and that is typically transitory. Eventually the patients will relapse. So a large part of the cost is that for 90% of the patient, you do a screen that is not likely to produce much of a result. So we need to do much better, and I think this is one of the areas where we're trying to get involved. We need to be much better in terms of A, having tests that cost less, and B, in terms of having tests that actually have make it valuable for a larger proportion of the population. So this is not to say that what we've done so far is not relevant. It's incredibly relevant. Just to give you a sense, the 10% of the patients that sort of have an advantage from precision medicine today, and it's 10% probably for immunotherapy and about 10% for targeted therapy, are more than what has been accomplished by smoke cessation, and that was a huge campaign. So we're talking about a major effect on cancer. Still, there's a big, and there's still a long way to go, because we want to go from that 10-20% to 100%. And so what we have done with our test is 100% based on RNA, and RNA is much cheaper to both isolate and profile. So the full test right now, which is available at Columbia, is below $1,000 cost in the CLIA lab, meaning that a fully CLIA compliant test that can be used clinically costs less than $1,000. So that makes it much more approachable. It also is a test that can be given as the tumor progresses, because while the mutations that accrue or that are selected as tumor progresses or responds with drug are generally a very poor indication of what the next line of therapy should be. Generally find mutation in KRAS or in P53 or other genes that are undruggable. The state of the cell, the RNA state of the cell, changes dramatically as you undergo therapy or as the tumor progresses, become metastatic, et cetera. And so you can literally use this test multiple times to assess what is the best therapy at each given point in time. Of course, this needs to be proven as having clinical value. So we're not saying that this test is the solution to cancer, but at least we're saying that we're excited enough by the results that we've achieved so far that it's worth testing these clinically on a large scale to see how valuable it can be.
So we have time for one last question. And so to wrap up, we were wondering, what's your outlook or what's your take on the future of systems biology and perhaps even synthetic biology? I know we spoke a little bit about systems biology before, but if you can relate the two also, like how is systems biology going to move forward, and if there's any association between it and synthetic biology and the future synthetic biology as well.
So that's a great question. I mean, so systems biology and synthetic biology are really two faces of the same metal, because the same way you think about, when you're studying a text, you go through first analysis and then synthesis, right? The system biology approach is the analytical approach that essentially identifies the fundamental principles that regulate the behavior of the cell, right? Once you learn those rules and you learn that some of them are amenable to a more engineeristic approach, where you could literally implement certain circuitry within the cells that are designed from scratch according to an intelligent design, which is intelligent because it's us, then that opens and paves the road to doing synthetic biology, which is essentially the implementation within the living cells of that particular circuitry. The example of how the two fields are really related is that some of the early work that was done by Jim Collins, in particular by Diego Di Bernardo, originally trained in his lab, was really relevant because they were able to implement a small circuit that had sort of embedded human proteins in a particular regulatory architecture within a yeast cell. And then, because the yeast cell is so easy to manipulate and grow, they could literally perturb the cells and generate tremendous amount of data that then could be analyzed by systems biologists to reconstruct the network topology. And so it was really an interesting effort where, in that case, an actual engineeristic design implemented in a cell was used to generate the data that the system biology could then use to reverse engineer the design itself. And I think a lot more of that is going to happen. Some of the principles that are emerging through systems biology are complex to implement from an engineeristic perspective because, for instance, we see an enormous amount of field effects where the individual contributors to the effect produce an extremely small contribution. But when you add them up together, then you have a strong contribution. So, for instance, when you look at height is the result of about 400 different SNPs or single nucleotide polymorphism in our DNA, each one having minuscule effect. We're talking about sub-millimeter effect. So when you add them up, you can change the height of a person from 4 feet to 7 feet, but individually the effect is very small, right? So the same thing happens in cells where you may have, and this is exactly how master regulator get dysregulated. You may have a protein that is critically relevant for a particular function, and yet that protein is not directly dysregulated by, say, a mutation or a variant, etc., but really through the input that it receives from hundreds of other gene products, including microRNAs, including upstream proteins, including all sorts of one network that we discover, which is RNAs regulating other RNAs through the shared microRNAs. And so you can have situations where there are very minimal dysregulation of, say, 15 different gene products upstream of a protein, and that protein now becomes extremely dysregulated. So that's something that's going to be challenging for synthetic biology to implement. But I think it could point out to general rules of how this kind of system works, almost like a neural network, and therefore the same way that we now design neural networks to drive cars, we may at some point be able to engineer neural networks that are implemented within living organisms.
All right. Thank you so much for your input, Dr. Califano. And that just about wraps up our podcast interview. And for the viewers and the audience, thank you for watching and tuning in, and hope to see you next time.